Thank you for that introduction. Everyone can hear me just fine. So let's begin. So the COVID-19 pandemic has probably been one of the more momentous events that we've had within our lifetimes. Okay, everyone can hear me clearly now? All right, so like I was saying, COVID-19 pandemic was probably one of the most disruptive events in our lifetimes, or at least mine, since I know a couple of you are more much older than me. Last night, using the Johns Hopkins web tracker, there have been over 650 million cases of COVID-19 worldwide, with 6.5 million deaths for what's going on. The pandemic, it was declared a pandemic back in May 12th of 2020, and it's still ongoing. I know there's been calls for it to say the pandemic has ended and COVID has become endemic, but it's still something that's a major concern, or at least influence for when it comes to policy and our lives. So due to this, our lab launched the creation of the coronavirus infectious disease ontology back in 2020. And we started out with the aim of having a systemic representation and knowledge representation of all the research that are going on for coronavirus infectious diseases. We were almost focused entirely on COVID-19. So when it comes to maintaining those ontologies, we try to preserve a couple of different principles. That includes the principle of minimal change and iterative ontology updates to minimize data loss. So that works well when we're in a domain that's already pretty well established, or something that isn't rapidly changing, but it becomes more complex when we're having this emergent situation with COVID-19. So previously, as part of our representation for the different coronavirus variants, we were focusing on the major clades that were popping up and the different naming schema that was going about. So we had GSI-8, which was the major source and collection of all the SARS-CoV-2 viruses, which were using their own nomenclature based off of very specific mutations that were found in each of the SARS-CoV-2 amino acid variants. We have the Pango Lineages, which is a more popular and widely used one, which uses their pangolin algorithm in order to find and reconstruct these lineages of how the virus spread, but that had some complexity since not all these viruses would share all the same amino acid variants, again, because it used a clustering algorithm to group them together, instead of just saying that these common amino acid variants means it belongs to this specific clade. The final classification is the one that the World Health Organization used in an attempt to harmonize and provide a controlled language for other groups to describe SARS-CoV-2 variants, and these are your alpha, your beta, delta, and omicron variants. So initially, we constructed the Has amino acid variant, which we would link up to a small series of initial amino acid variants, which we defined as individual residues or mutations that were we have been known to be either characteristic or associated with specific viral variants. And of course, each of these are then in turns linked to an appropriate protein. The earlier definition of coronavirus amino acid variant has since shifted, and which I'll discuss more, but I just want to show what we had historically before we're doing this round of updates. So one of the concerns we have when it comes to representing these terms of ontology is the idea of variants of interest and variants of concern. And these are specific SARS-CoV-2 variants that have been specifically identified by the World Health Organization or other groups as specific lineages that we need to monitor because of their potential for health concern, in the case of interest, or if they have been shown to cause negative effects in viral transmission, or have increased rates of immune evasion, or otherwise have a serious impact for human health. So while we wanted to represent these terms ontologically, it's not that useful since these terms fluctuate based on if they're currently something we need to worry about. So alpha, beta, and gamma variants, for example, were part of the initial set of 4 variants, designated variants of concerns, but they have since been no longer relegated as variants of concerns since they are functionally extinct. In addition, while we have these schemas and different lineages for their names, SARS-CoV-2 is still evolving. So while we have already tried to harmonize and put relationships between variants, these actually clades are not necessarily the same from when we first identified them in 2021 as they are today in 2022, or how they're going to continue to be in the future. So right now we have the Pango super lineage, and I'm showing all of Delta in the orange color, while all of Omicron in the blue color. So while if you were to query it ontologically, you would see assertions that these terms are equivalent to each other or match up. However, the next string, naming schema, goes off whenever there's been a sublineage that is distinct enough to merit grouping into its new classification. So while at the beginning you would only have Omicron as 21K, it has since diverged into 21L, 22A, 22V, into a wide variety of different clades, which makes this somewhat difficult to keep consistent ontologically, because it's not necessarily the same besides a shared heritage or inheritance. Again, I mentioned earlier that pangolin, which is where we're using the initial message, are focusing on the actual naming schema for the lineages, and that they currently have a very nice web tool, which if you go add to query, it will show you all of the characteristic amino acid variants for the SARS-CoV-2 viruses have at this time. And while that's very convenient if you want to, say, run a query today or for what's going on, that also means since we're constantly getting new sequences added, we're getting some of the percentages change. That means over a period of time, we're going to see this type of semantic drift in what the actual definition of Omicron or what Delta variant is actually going to be for the virus. So we're having some of these definitional changes going on in the naturally semantic drift going on, but we also want to try to minimize the changes to our ontology as a matter of keeping it consistent and interoperable. So the question is, what's the best way to account for the increase of new information we have for all the amino acid variants, while also not doing drastic changes to our core hierarchy in ontology? So earlier on, in addition to the viral variants, I brought up we had specific examples of amino acid variants as terms that we can help annotate and link to the viral variants. So using current definition of amino acid variant as a chemical entity, that is an amino acid variant of a coronavirus proteome, which strictly speaking, all the terms are green are terms currently in CITO, since we don't have the amino acid variant specific as a CITO-specific term, but the one that we've been using in development, or at least the one that I've been using in development, is that an amino acid variant is a chemical entity that is a set of amino acid residues that differ in reference to some protein. So very small specific changes. By using these definitions and tracking the effects of these residues, we can provide a consistent, or at least query or comparison across multiple different SARS-CoV-2 viral variants or viral sequences to whenever we want to do comparison or integration of the actual results. So I also provided a small definition of mutation over here as being separate from variant. And part of that relates to how we define a variant as an existing set of amino acid residues. But there's a couple noteworthy amino acid variants or mutations which end up as deletions, which due to how we assert it no longer makes sense with that definition. So we ended up having to use two definitions, one to cover the actual deletions or nonsense mutations, those that have been a single substitution or actually leave an existing residue. So as a way to help keep myself straight and make it clear, we have been mostly focusing on the small chart. Yeah, go ahead. Yeah. Okay. Are we recording? Oh, sorry. So while we have the CITO-specific terms that are shown in brackets here, I've also listed individual variants of interest and variants of concern. Those are not CITO-specific terms, but those would cover alpha, beta, delta, and gamma, and all of those I will refer to as viral variants. For the coronavirus amino acid variant refers to specific amino acid residues that differ within the SARS-CoV-2 proteome. And I'll be using that as matter of consistency for currently in the ontology in CIDO when we're using variant for referring to residues. Otherwise, as part of the specific process for the coronavirus mutations, we will refer to them as coronaviral amino acid mutations, either in the case of deletions or for nonsense mutations. So I'm just doing this as a way to make clear, since a lot of these words sound familiar and will drag together. Okay. So when it comes to collecting all the amino acid variants as part of the update to CIDO, this was mostly done through manual collection of all the listed websites and web tools that are available for amino acid variants. So in addition to Pango, which is doing the dynamic one, they also have outbreak.info, which serves as their primary hub for the individual amino acid mutations for a specific Pango lineage. GSI-8 also had a collection, or previously had a collection of amino acid variants that they did for alpha, beta, and gamma through they've stopped updating since then. I also paid attention to covariance.org and Lineages, both of which are good resources that have been keeping track of both experiments and the effects of these individual mutations. Finally, when it comes to actually taking a look at the specific domains or any other annotation features where have been going on, I've used the above resources and the literature survey to look for experiments to see any other changes or for both domain inclusion and for the effects that they have and the effects of variants have on SARS-CoV-2 or COVID-19 disease process. So as part of the framework for what's going on, we have the setup for the ODP for SARS-CoV-2. Currently, A covers the SARS-CoV-2 amino acid residue and variant and represents a bit more complex hierarchy, for it's still relatively simple. So we said that each part of the amino acid variant is part of a specific protein domain, and then it's based off a central hierarchy of amino acid variant in SARS-CoV-2 protein or amino acid variant in SARS-CoV-2 protein domain. We distinguish that as part of the central hierarchy because it makes it somewhat easier to add annotations for common features that we notice when doing part of the SARS-CoV-2 analysis. We have also included processes for individual features or to show changes in the amino acid variants for the use of susceptibility eligibility process, or more specific terms such as susceptibility decreases vaccine efficacy against, which again we do as part of all based off of experimental evidence. But the core part for each of the amino acid variants is primarily based off of asserting that each amino acid variant has a specific amino acid position and has mutated from an initial amino acid residue and now exists as having part of a new amino acid. The structure for SARS-CoV-2 amino acid mutation is a bit simplified, again, because this is a process. And we mostly have the specified input of the initial amino acid having some amino acid position, while the actual mutation event, either deletion or nonsense, is already asserted as part of the class. Okay. So showing off this primary use case of ODP, I'm going to focus on SD614G, which is probably the most famous of the different amino acid variants. It was one of the first ones to be experimentally verified and has been shown to have different effects when it comes to viral transmission, binding to human ACE2 S binding, in addition to basically involved at almost every step of the process for the actual viral infection of SARS-CoV-2. So as you can see, we have listings for specific characteristic amino acid variants, in addition to listing as part of the amino acid position, and of course, with what we have shown up as part of the literature. Whenever we have a detail about affecting the specific process that it's binding to, either as human H2 entry, which is the one that mediates the binding to the specific host, as part of the ribosomal binding membrane, versus individual effects, which we can be more specific as part of the amino acid variant. So one thing that I'm bringing up is that for D614G, you notice that we have characteristic amino acid variant of some Omicron variant. And as part of the way to handle the different variety we have for each of these websites having slightly different definitions, either due to using a different algorithm to predict similarity or just doing so at a different timestamp. So we distinguish between is amino acid variant and is characteristic amino acid variant. So the former is done whenever we see amino acid variant as being called as associated or already linked to a specific SARS-CoV-2 viral variant. Characteristic is a subclass of is amino acid variant, where we deal with a situation where we're saying that the presence of a specific amino acid variant is used to identify specific SARS-CoV-2 as belonging to a specific viral variant. So, okay. I think this is an older version, but. So here we have a representative example of some of the SARS-CoV-2 amino acid variants that are showing up. And we have a consistent naming schema that we handled as part of the ontology. So each of the amino acid variants are provided with a short form of including the initial protein name, followed by the reference amino acid or the amino acid found in the wild type SARS-CoV-2, followed by the amino acid position that where this mutation or variant exists, along with the resulting new amino acid if it's just a residue, or what type of mutation event that's going on. So you can see SD614G as representative here, being listed along the protein domain, which is where it's located in the central hierarchy for the amino acid variant, along with the presence of which viral variants were associated. Okay, I think this is an older version, so I apologize, but what I wanted to highlight over here is that one of the amino acid variants over that is the nuclear capsid RG203KR. So this is more of a legacy amino acid variant in that we have two specific mutated amino acid residues that are strongly associated with each other as part of experimental evidence. So while those still exist as part of the initial version, Ontology, it also has two child classes, which follow the normal framework of NR203K and NG204R. So we can still accommodate older versions or clustered amino acid variants by treating it as part of amino acid domain, and doing part of the annotation schema there, or we can just focus on more specific child classes. Okay, I think this is an older version of my PowerPoint, so I apologize. So what is currently missing from this presentation is mostly a focus on the specific subdomains and the frequency for the amino acid variants. So the most common amount of amino acid variants occurs with an S protein, which is about 62% of all the amino acid variants currently in SIDO. Most of these tend to be characteristic in a few individual viral variants, where the largest cluster is with Omicron, which has 28 of these specific effects. The second largest one happens to be in the polyprotein 1AB, which contains 28% of the current variants in SIDO. And this protein is responsible for changes in viral replication, viral assembly, and split into a variety of subdomains that are linked through there. So when we're getting into the central hierarchy for each of the amino acid proteins, we take a look at the subdomains. So the S protein has two major subdomains, S1 and S2. The S1 domain is defined through text definition of how many amino acid positions they are located within SARS-CoV-2. So we have the initial S1 and this can be overlapping. So we have the receptor binding motif, which is contained inside the receptor binding domain, which is contained within just the normal S1 domain. And each one of these processes have been shown linked through experiments in the literature to specific processes that affect the COVID-19 disease process. Now, some of these can also be linked to changes when it comes to drugs or vaccines. such as the N-terminal domain as being used for the affinity to the treatment delivered. The S2 domain, while being less interesting in terms of experimental response, since most vaccines and drug treatments focus on S1, still plays a variety of roles that affect the actual viral protein stability, which is important for how the natural protein will be able to interact conformationally with human ACE2 and actually enter into the cell. You can still see we have a larger amount, even though it tends to be more spread out through individual variants, which are shown on the left side, along with mutations, which are on the right. Okay. Yeah, this is an old version of my PowerPoint, so this got fixed. So one of the identification of the amino acid variants for part of the subdomains was used through a DL query, where we take a look for characteristic amino acid variants that are unique to the ribosomal binding motif region within S1. Since this motif is the one that directly binds to human ACE2 and initiates viral entry to the cell, we were mostly interested to see if there were a common set of highly influential mutations that are already found, since almost all of the characteristic amino acid variants that are found to identify the variants of concerns were set up in such a way we expect these to have very high impact. And when we did do that query, we found that the literature confirmed our results, with each of these proteins having very specific experimental evidence of increasing either viral transmission, increasing immune escape, or other healthcare-related effects. So now that we took a look at, we used our DL query for the actual domains that we have set up and see if we can identify further annotations or results for the amino acid variants. We then took a look at the opposite question, which is if we take a look at our existing amino acid variants and see if they have a shared specific experimental associated function, can we also use that and expand whatever definition or functions we've found and annotate or provide some guidance for research here. So while some of these are relatively well established, so increased viral transmission due to changes in NTD domain, or immune evasion from RBD domain. We also found that there was a cluster, or as part of ORF9b, which is currently not annotated as part of UniProt, that deals with our autophagy suppression as part of the actual immune expression. So using this, we got an additional set of domains, or at least regions, which we can then further suggest to UniProt to guide as further research or annotations for new and novel biological functions currently not included there. So as part of the future directions for that, we're focusing on the paper, one of which was integration of our results into epitopes as part of IEDB, since we're expecting SARS-CoV-2 mutations for those specific results to end up having those effects. And then we also want to take a look at experiments of resolving integration of multiple amino acid mutations together, along with, since so far we only looked at the effects of individual mutations within its domain, even though that these domains end up creating a conformational structure which can be changed. While unlisted, we also had some earlier terms in CITO that were developed that would be more appropriate as part of vaccine ontology, such as susceptibility decreases vaccine efficacy. So realignment some of those terms to apparent ontology is another further guidance. So in conclusion, we mostly updated CITO to include an additional 130 amino acid variants and added associated axioms and relationships for them. We provided an ODP for the inclusion of additional amino acid variants for SARS-CoV-2, which we can then use for any future advancements. We provided additional quantitative annotation for some novel amino acid domains that can be done to UniProt or guide further research when it comes to SARS-CoV-2 related vaccine work. And we also provided specific processes or domains that we can use to predict the effects of individual mutations or future amino acid variants of SARS-CoV-2. Okay, I also thank everyone for Any questions? I also apologize. Looks like an earlier draft of my PowerPoint got uploaded instead of the most recent one. So I apologize for the odd content towards the end.
If there's no question. If I can ask a question, so can you elaborate how we can use the CIDO with the amino acid and variant information incorporated? How can we use this resource for further kind of vaccine research or efficacy research?
Okay.
And can you repeat the question?
Yeah, so, okay, this is working good. So the question was, how can we use CIDO in order to further vaccine research when it comes, especially with the integration of amino acid variant tools? So each of the amino acid variants have a specific amino acid position, which we can then use and align to as part of IADB existing epitopes that we know are already part of existing SARS-CoV-2 vaccines or predicted as part of any future vaccines that use SARS-CoV-2 spike protein as an example. So we can align the specific amino acid positions with the known positions of immune epitopes for SARS-CoV-2 vaccines. And we can see, first of all, will this mutation end up having a change for the epitope, even if it's just simple substitution? And if so, if it has substitution, we can take a look at confirmation and then see if that ends up affecting its reactivity or recognition sites. So this is more of a future direction thing since we didn't do a query for the specific epitopes, but by matching up the position of these amino acid variants to known SARS-CoV-2 viral epitopes, we can use that to predict if we're going to see a drop or decrease for vaccine efficacy. Okay.
Thank you. Do we have any question or any questions from the online audience?
Yeah, it's great.
If not,